So now we know how to make nucleotides and what drugs target those pathways. So now let's go over how nucleotides are broken down. For the step 1, it's most important to know about how to break down purines. Now a lot of this overlaps with what I just went over, but I'm going to rearrange the layout a bit so it looks more like what you have in the book. Alright, so remember, to make purines, we start by adding hypoxanthine to PRPP to make IMP, or inosine monophosphate. One thing I didn't tell you before is which enzyme catalyzes this reaction, and that's HGPRT, which stands for hypoxanthine guanine phosphoribosyltransferase transferase. A nice thing about biochemistry is that the enzyme names generally tell you what they do. So in this case, we're transferring a hypoxanthine onto a ribose that has phosphates on it, which is PRPP. IMP can now be converted to either AMP or GMP, as I talked about in the last slide. But what about the G in HGPRT, which is guanine? HGPRT can do the same thing with guanine that it does for hypoxanthine, to make GMP. However, you need a different enzyme to use adenine to make AMP, which is APRT, or adenine phosphoribosyltransferase transferase. Alright, so sometimes you don't have enough nucleotides and you want to make more, such as in a cell that's rapidly dividing, but sometimes you have too many and you want to get rid of them. So how do we break these guys down? Let's start with GMP. GMP can be broken down into guanosine, and do you remember the difference between GMP, guanosine, and guanine? So here's GMP, which, as the monophosphate in the name suggests, has one phosphate, and it also has a sugar and the base. If we remove the phosphate, which is what we're doing here, we're left with guanosine, and then if we remove the sugar, which we're doing here, we just have the base, guanine, left. We can do basically the same thing to get from IMP back to hypoxanthine through inosine. Although AMP doesn't get converted back to adenine, instead it's converted to hypoxanthine through an inosine intermediate. This requires the enzyme adenosine deaminase, or ADA, since we need to deaminate adenine to create the hypoxanthine base. When ADA is deficient, adenosine begins to accumulate and feeds back to inhibit ribonucleotide reductase, which is the enzyme that produced it. As you might remember from a few minutes ago, ribonucleotide reductase is required for the synthesis of all deoxyribonucleotides. Since it's inhibited in the case of ADA deficiency, nucleotides can't be produced, which mostly affects cells that need to divide a lot, such as those in the immune system. Therefore, patients who have ADA deficiency will have a low lymphocyte count and can also have severe combined immunodeficiency, or SCID. Okay, we're almost done with this. So now that we've converted everything into hypoxanthine and guanine, we can use the enzyme xanthine oxidase to convert them first into xanthine and then into uric acid. What happens to uric acid? It's excreted in the urine. So now that we've got this whole pathway laid out, let's talk about what can go wrong here. I already mentioned how ADA deficiency causes immune disorders, but what happens if a patient is deficient in HGPRT? They won't be able to make GMP or IMP. So instead, guanine and hypoxanthine will accumulate and then be broken down into uric acid. Accumulation of uric acid can cause hyperuricemia and gout, and indirectly cause some neurological problems, including mental retardation and self-mutilation, which often includes nail biting, aggression, and chorioathetosis. What's this syndrome called? This is Lesch-Nyhan syndrome. HGPRT is on the X chromosome, so this is an X-linked recessive disease. And you can remember that this gene is involved in purine salvage pathways using the mnemonic, "He's got purine recovery trouble." Okay, so what can you do to treat this? One commonly used drug is allopurinol, which inhibits xanthine oxidase here and here. This will decrease the amount of uric acid in the blood. Now here's an interesting pearl for you. Xanthine oxidase metabolizes normal purines, but can also metabolize drugs that mimic purines. One example I mentioned a couple minutes ago is 6-mercaptopurine. Do you remember what that's used for? 6-mercaptopurine is an anti-neoplastic, which is basically a modified purine, which binds to and inhibits enzymes that normally synthesize purines, thus inhibiting purine synthesis. And it's used to treat leukemias and inflammatory diseases like rheumatoid arthritis and inflammatory bowel disease. So let's say you have a patient with Lesch-Nyhan syndrome who you're treating with allopurinol. And this patient is also now diagnosed with a neoplasm that needs to be treated with 6-mercaptopurine. What do you think would happen? Since xanthine oxidase normally metabolizes 6-mercaptopurine, but the allopurinol is inhibiting xanthine oxidase, the patient will not be able to metabolize 6-mercaptopurine as effectively, and therefore needs a lower dose of it. Next, let's talk about the genetic code. I won't spend a lot of time on this since you probably learned about it in high school, but I'll cover some of the key points that you might see in your exam. So the genetic code is what is used to translate DNA, or more directly RNA, into proteins. This is a codon table, and it shows that each amino acid is encoded by sets of 3 nucleotides called codons, and many amino acids are encoded by multiple sets. For example, proline is encoded by CCU, CCC, CCA, and CCG. This means that the code is degenerate or redundant, since multiple codons can code for the same amino acid. You might notice that of the amino acids that have multiple codons, the codons usually only vary by the last nucleotide, which is why this is called the wobble base. Mutations in these third nucleotides will often not affect the amino acid sequence of the protein, and therefore these are often silent mutations. Another feature is that the code is unambiguous, which means that each codon only codes for one amino acid. Our third feature is that it's commaless or non-overlapping. This is easier to explain with an example. So let's say this is part of an RNA sequence that's being read by a ribosome. After each codon is read, the ribosome moves over by 3 nucleotides, so it reads this one, and then this one, and then this one. No overlapping parts of codons will be read, like this, and no nucleotides will be skipped. And the only exception here is that some viruses can read RNA as overlapping codons. The last feature is that the code is universal. This means that in all species the code is the same. If a codon codes for glutamine in one species, it'll also code for glutamine in another species. An exception to this is in mitochondria, which have their own tRNAs, which in some cases match different amino acids to codons than a nuclear encoded gene. While we're here, I also want to point out the 3 codons that don't code for any amino acid, but signal the ribosome to stop, and these are called the stop codons, which are UAA, UAG, and UGA. These are the only 3 that you might want to memorize. Now that we've talked about what DNA is made of, the nucleotides and codons, let's go over how it replicates itself when cells divide. Can you think of some problems that cells need to consider when replicating their DNA? Cells need to make sure you get exactly 2 copies which are identical. They have to make sure replication happens only once per cell cycle, which is during S phase, and that everything happens quickly enough that rapidly dividing cells won't be held up while they wait for DNA to replicate. After all, you've got about 3 billion base pairs in the cell. Cells also have to minimize mistakes, such as single nucleotide changes or expansion of repeated sequences, and they have to fix mistakes that occur. And lastly, they need to deal with the issue of not losing DNA at the ends of the chromosomes, which I'll get back to in a minute. Alright, so where do we start? Well, DNA replication starts at an area called the origin of replication. How many of these are there? Well, there's only one in a prokaryotic chromosome, but eukaryotes have a bunch of them since they have a lot more DNA, and it would take forever with only one. At this origin of replication, the 2 strands of DNA separate and forms what looks like a bubble. Each end of this bubble is the replication fork, and as replication progresses, this will expand outward as the new strands of DNA are formed. Let's take a look at one of these replication forks. So the blue is the parent DNA, and I'll show the newly synthesized DNA in red. The 2 strands replicate in slightly different ways. One replicates continuously, called the leading strand, and the other replicates in pieces, called the lagging strand. Now why can't the lagging strand create its new daughter strand continuously? Well, this has to do with the polarity of DNA. Do you know which end of each strand here is the 5' and 3' end? You might want to pause the video and think about it for a second. So DNA is always synthesized in the 5' to 3' direction, and that's referring to the direction of the strand being synthesized, not the original. On the leading strand, this new piece of DNA can be synthesized continuously, since the replication fork that it's going towards is at the 3' end of the strand being synthesized, and therefore the origin of replication is at the 5' end. However, the lagging strand has to be synthesized in the opposite direction that the replication fork is moving. So as the replication fork progresses, it has to keep starting over. Do you know what each piece of DNA in the lagging strand is called? That would be an Okazaki fragment. Okay, let's look at some of the enzymes involved. I'll mostly go in chronological order. The first is helicase. So what does helicase do? It unzips the DNA and allows the bubble to expand. It's actually a little known fact that helicase is the most fun of all the enzymes since it spends all day unzipping genes. Get it? Sorry, bad joke. Anyway, helicase does this just by disrupting hydrogen bonds between the 2 strands. It doesn't make or break any covalent bonds. Since helicase is moving along with the replication fork, something is needed to prevent the strands from reannealing to each other, which complementary strands of DNA like to do. Do you know what protein does this? This is where a protein called single-stranded binding proteins comes into play, which is just that, a protein that recognizes and binds to single-stranded DNA to prevent it from binding to its complementary sequence in the other strand. The last proteins I'll mention on the subject of opening DNA up are the topoisomerases. These are proteins that regulate supercoiling of DNA. An analogy you can use to think about this is think of two strands that are anchored at one end, but are then wrapped around each other a few times. If you pull the two ends apart, they'll be wrapped tighter and tighter until pretty soon you can't pull them apart at all anymore. This is why you need topoisomerases. Now, regulating supercoils is a vital process in cells, so if you inhibit them in bacteria using fluoroquinolones, the bacteria are unable to divide. Alright, back to synthesis of DNA. So what enzyme is in charge of synthesizing the leading strand? That would be DNA polymerase III, which synthesizes the leading strand continuously towards the replication fork. Once again, always 5' to 3'. Okay, so one more thing that needs to be done before we can make the lagging strand is adding RNA primers. There are no DNA polymerases that can initiate DNA synthesis from scratch. They have to build off of existing DNA or RNA. So what enzyme can help us here? That would be primase, which can synthesize RNA from scratch, by which I mean that it can add ribonucleotides to their complementary DNA bases and connect them together. Once a primer has been laid down, our friend DNA polymerase III can jump in and start adding deoxyribonucleotides to it, just like it did for the leading strand. DNA can only be synthesized in a 5' to 3' direction. So DNA polymerase III starts from the RNA primer and builds towards the 3' end of the new strand. So as DNA polymerase is making each new strand, it proofreads its work, and if it realizes it made a mistake by adding the wrong nucleotide, it can remove it by using its 3' to 5' exonuclease activity. This is of course the opposite direction of DNA synthesis, since it's going backwards to remove a mistake. After the lagging strand has become a continuous stretch of alternating RNA primers and Okazaki fragments, the only thing left is for a different polymerase, which is DNA polymerase type 1, to remove the RNA primers using its 5' to 3' exonuclease activity, and fill in the gap with DNA as it goes. Polymerase 1 has the same synthesis and exonuclease functionality and polarity as polymerase 3, but has the added ability to remove RNA primers. Alright, so one last enzyme here. DNA ligase will seal the hole between the DNA fragments. Keep in mind, this is between the fragments of the same strand of DNA, not connecting one strand to its complementary strand in a double helix. The last problem we have is what to do about the ends of the chromosomes. The leading strand is no problem since it can just synthesize DNA all the way to the end, but the lagging strand is dependent on RNA primers, and the last RNA primer can't be filled in with DNA once it's been removed, since DNA synthesis would require another primer to build off of. Therefore, a few hundred nucleotides of DNA are lost each time the cell divides. The solution is to use telomerase, which is a ribonucleotide protein that repetitively adds a 6-nucleotide sequence, TTAGGG, to the end of each lagging strand, creating non-coding junk DNA that can be lost without harming the function of the cell. In adults, telomerase is only expressed in cells that need to divide a lot, such as stem cells and some cells in the immune system. Although mutations that can cause telomerase to be expressed in other cell types are one of the things that are necessary for neoplastic transformation, since without telomerase, even cancer cells will lose the ability to divide after a while. Alright, let's do another flash quiz. So, what symptoms would you associate with a deficiency in the enzyme HGPRT? This is Lesch-Nyhan syndrome, which has the symptoms intellectual disability, self-mutilation, aggression, gout, and dystonia. For bonus points, how would you treat this? Well, the treatment here is allopurinol, since it inhibits xanthine oxidase, preventing uric acid formation.